So in the third and final part of lecture 4, I talked about the COX enzymes, which are inhibited by NSAIDs. And of course, this is an example of an undesirable effect, and the use of NSAIDs is an important cause of gastric ulceration. In fact, there are two COX enzymes, COX-1 and COX-2. And these enzymes are really very similar, both at the amino acid level. So if you compare amino acids at a certain position, they're often identical between COX-1 and COX-2. And as you expect, perhaps based on that observation, the structure of COX-1 and COX-2 is also really very similar, as you can see here in these diagrams here below. So this is COX-1. The structure of COX-2 is here in purple, and you can see that they're really very similar if you overlay them on top of each other. So these COX enzymes, COX-1 and COX-2, are both involved in the formation of prostaglandins using arachidonic acid as a substrate, and they're attached in the cells intracellularly. And so they're located at the membrane of the endoplasmic reticulum, where the arachidonic acid normally resides. Now, if we look in a bit more detail at the active site of the enzymes, so where the catalysis is taking place, you can see here the arachidonate in grey, shaded in grey. And the arachidonate contains a carboxylate group that interacts with an arginine residue of the COX enzymes. And as you may remember from case 3, many NSAIDs contain a carboxylate group that interacts with this very same residue. Serine 530, highlighted here in red, is covalently modified by aspirin. And NSAIDs contain, in addition to this carboxylate group, as you may remember from the other lectures from the chemistry and case study 3, they contain, in addition to this carboxylate, aromatic ring structures that can interact with other aromatic rings. For instance, the tyrosine residue of the COX enzymes indicated here. So NSAIDs work by interacting with the arginine, the serine in the case of aspirin, and this tyrosine. And so basically NSAIDs block the binding site of arachidonate, and so if the NSAID binds to the COX enzymes, arachidonate cannot bind at the same time. Now, another characteristic of the COX enzymes is the fact that they require a cofactor. So here's the heme group, this iron with this porphyrin group, and so the COX enzymes are yet another example of an enzyme that contains a cofactor for its function. So while the COX-1 and COX-2 enzymes are very similar and carry out basically the same reaction, there are some unique characteristics of these enzymes. So COX-1 is a constitutive enzyme, so it's present in many cell lines at a more or less constant level. And COX-1 is important for various organ functions. So it's important for renal function, and of course it's very important for the protection of the gastric epithelium, because the prostaglandins stimulate the gastric epithelial cells to secrete mucus. And so it's the undesirable inhibition by NSAIDs that causes the the defects in the gastric protection, leading to gastric ulceration. Now, COX-1 was known for many years before COX-2 was discovered. And soon after COX-2 was discovered, people found that this enzyme is inducible. So it's normally present at very low levels. But in inflamed tissue, this enzyme is really induced to fairly high levels. And so people realized after this discovery that perhaps the inhibition of COX-2 in inflammatory tissue is mediating the desirable effects of NSAIDs by reducing inflammation and by reducing pain, and that the inhibition of COX-1 was undesirable, resulting in the adverse drug reactions. And so this insight into the biology of COX enzymes then sparked an interest to search for compounds that could selectively block the COX-2 enzymes. And so this is an example of rational drug design or rational drug discovery, which means the discovery of drugs based on biological insights of the targets. So how can we understand selective inhibition of the COX-2? So to understand this, I just made these diagrams that just take into account 3 amino acids of the COX-1 and COX-2 enzymes. And so in COX-1, these are 2 isoleucine residues and a phenylalanine. And in COX-2, the amino acids are slightly different but very similar. So they contain 2 valines instead of isoleucine and a phenylalanine. And as you can see here in this diagram below, the valine residues found in COX-2 are slightly smaller than the isoleucine residues found in COX-1. So this pocket in COX-2 is slightly larger than the pocket, the comparable pocket in COX-1. Now you can also see that a typical non-selective NSAID inhibitor, so this one can inhibit both COX-1 and COX-2, is smaller than a selective COX-2 inhibitor, shown here in red. So in case of a non-selective inhibitor, here in blue, this compound can bind both to the COX-1 and COX-2 enzymes. So you can see there's a bit more space, a bit more room in the COX-2 enzyme. But both in COX-2 and COX-1, this non-selective inhibitor can bind. Now, by contrast, a selective inhibitor, such as this compound SC558, which is shown in red here. So this is a much larger compound, a bulkier compound. And as you can see in this panel on the right, this inhibitor can bind just in the pocket of the COX-2 enzyme. But in case of the COX-1, you can see that there is no room for this larger selective COX-2 inhibitor. You can see it clashes with some of the side chains, so there's basically no space for this selective inhibitor to bind to the COX-1. But because the pocket is slightly larger in COX-2, there is room for the selective inhibitor to bind, and it will inhibit COX-2 but not COX-1. And so here's just another view of the same situation. So just another angle, you can see on the right that this selective COX-2 inhibitor just about fits in the pocket of the COX-2 enzyme. But in case of COX-1, it clashes, so there's no room for this compound to bind in the COX-1 enzyme, and so it will not inhibit COX-1. So when the first selective COX-2 inhibitor came onto the market in the US, they were really successful. They became the fastest selling drug in the history. However, after initial questions about cardiovascular safety, it was widely debated whether there was an advantage of using selective COX-2 inhibitors over the combined use of just NSAIDs in combination with gastroprotective drugs such as the proton pump inhibitors. And finally, it became clear that even selective COX-2 inhibitors are not fully free of adverse reactions concerning the gastric epithelium. And so, for a combination of reasons, selective COX-2 inhibitors have not caught on as much in the UK and Europe as compared to the US, perhaps. However, the story of selective COX-2 inhibitors demonstrates the process of rational drug design or rational drug discovery, and it illustrates the potential and the problems of this approach. So the potential in terms of the selective targeting of highly related proteins such as COX-1 and COX-2, but also it highlights the problems with this approach, and that is the complexity of the underlying biology leading to unanticipated effects which are illustrated again by this story.